Hi everyone, my name's Raven, and today I'll be talking to you about my research on how urbanization influences isotope signatures and antioxidants in eastern chipmunks. Before I get started, I'd just like to say thank you to my supervisor, Dr. Albrecht Schulte-Hostedde, for giving me this opportunity to conduct my research, as well as my two committee members, Dr. Jackie Litskis and Dr. Mary Martinez, for their awesome feedback so far, as well as a big thank you to everyone that allowed me to trap chipmunks in their backyard this past summer. As a grad student, I like to procrastinate doing my research by spending time on Instagram. And one day, while I was on Instagram, I came across an account for an individual named Van Gogh, and I'm not talking about the artist. I'm talking about the chipmunk. Van Gogh is a chipmunk that's hand-fed by a woman named Allie. Not only does Allie feed Van Gogh, but she also feeds Mona Lisa and Jackson Pollock. And I'm sure many of us can relate to Allie in the sense that at some point in our lives, we've decided to feed an animal because we thought it was cute. Not only do animals get access to our food through direct feeding, they also get it through bird feeders. But the most important access point for human food waste to animals is through our garbage. And with urbanization, or the growth of cities occurring across the globe, animals have more and more access to our food waste every day. Prior research has found that chipmunks living in cities have lower stress hormones than their natural counterparts, presumably because of the dietary differences that exist between habitats. So, for example, a chipmunk living in an urban environment likely has access to readily available human food waste, which means it can forage less and perhaps has less natural predators within an urban habitat, so it doesn't have to worry about being attacked while foraging. However, how different the diets are between an urban and a natural habitat is unknown, and there's two different ways that we can determine what chipmunks are eating in urban and natural habitats. The first way is using isotopes. So isotopes are essentially elements like carbon-13 and nitrogen-15, which may differ between urban and natural habitats. Carbon-13 is representative of a corn-based diet, so in human food we typically consume a lot of cornstarch or corn as a filler, and if we're throwing that out in our garbage or putting it into our bird feeders, it's likely chipmunks in urban habitats have access to it. Nitrogen-15 is representative of protein, so if we're putting out lots of seed into bird feeders, or perhaps throwing out a lot of meat waste, chipmunks may have access to that as well in an urban habitat. We can also use antioxidants. Antioxidants are found within our fruits and vegetables and are important for reducing oxidative stress. Oxidative stress occurs when too many free radicals are present within mammalian systems. So a free radical is essentially a molecule that likes to bind to proteins and enzymes and inhibits its function, which is bad because that's been linked to many different diseases such as diabetes, obesity, and cancer. Antioxidants work to stop free radicals from binding to those molecules by binding to them first. So it's possible because chipmunks living in urban habitats have access to our vegetable waste through compost bins and garbage bins, they may have a lower risk of oxidative stress because they may have access to more fruits and vegetables than their natural counterparts. So based on this idea that chipmunks may be consuming different diets between urban and natural habitats, I wanted to investigate whether chipmunks in urban habitats consume more corn-based and protein-based foods and more antioxidants than chipmunks in natural habitats. My first prediction is that chipmunks in urban habitats will consume more antioxidants compared to chipmunks in natural habitats, again due to the access of compost bins and garbages. And my second prediction is that chipmunks in urban habitats will have higher carbon-13 and nitrogen-15 signatures than chipmunks in natural habitats because corn-based food waste and protein-based food waste are readily available in urban habitats. I chose to study chipmunks for my research because, as prior studies have shown, chipmunks are fat and happy in the city because of those stress hormone differences that we see, which suggests that there's some sort of physiological consequence of living in an urban environment, which may be because of diet. As well, chipmunks are abundant in both urban and natural areas of Sudbury, which make them ideal to get a large sample size. Throughout this past summer, I collected samples from 115 chipmunks throughout 20 study sites in Sudbury. Each time I caught a chipmunk, it was given a metal ear tag like the one you can see here, which had a unique number on it, so if the chipmunk was recaptured throughout the study, I could collect new samples from it. I collected fecal samples from each chipmunk, which allowed me to determine the amount of antioxidants that they were excreting using a total antioxidant capacity assay, which is essentially a colorimetric test that tells me how many antioxidants are within a sample. Hair samples were collected from the rump, just above the tail, and sent to Dr. Hobson at Western University to determine the carbon-13 and nitrogen-15 concentrations within each sample. Next, I had to determine how urban or natural my study sites were. In urban ecology, most studies define urban and natural habitats categorically, rather they should be defined as a gradient. So in order to do that, I conducted surveys and used GIS. At each of my 20 study sites, I visited them for 3 days each, and every hour from 7 a.m. to 1 p.m., I counted the number of dogs, people, cars, and bikes that passed by every hour, as well as the number of trash pieces, trash cans, and bird feeders found within a 100-meter radius of that trap line. At the end of that 3-day period, I took the average of all of those variables. Next, I determined the percent of vegetation cover at each study site using the Canada Land Cover Map in GIS. And taking those two components, I put them into a principal component analysis to determine the natural urban score for each of my study sites to generate the gradient. PC1 was representative of the natural urban score. So all the variables that I measured had these factor loadings on PC1. You can see that people had the heaviest loading where vegetation cover was the opposite, suggesting that people was the most urban factor, whereas vegetation cover was the most natural factor. And PC1 explains 44.9% of the variation within my data set. Next, we can visualize the gradient of my study sites. So on the x-axis, those are my study locations, so all 20 of them within Sudbury, in relation to PC1, which represents the natural urban score. A more positive number represents more urban habitat, where a more negative number represents a more natural habitat. And this is what I found. So I had a pretty even split of urban and natural study sites between the Sudbury city limits. And you can see that PC1 again explains 44.9% of the variation within my data set. Next, we can talk about carbon signature results. On the x-axis, I have my natural urban score, or PC1, and a more negative number represents a more natural habitat, where a more positive number represents a more urban habitat. And on the y, here are my carbon values, and this is what I found. So there is no effect of urbanization on carbon-13 signatures based on that p-value. Next, we can consider nitrogen. So again, on the x-axis, my natural urban score in relation to the nitrogen-15 values, and this is what I found. You can see that the p-value is less than 0.01, which means there was a significant effect of urbanization on nitrogen-15 signatures. For antioxidant concentrations of the fecal matter across the gradient, on the y-axis here we have the total antioxidant capacity of the fecal matter. I found that there was pretty variable results in the fecal matter, and there was no significant effect of urbanization on the antioxidants in the fecal matter. Just to summarize my findings, urbanization had an effect on nitrogen signatures, which was reflective of protein, where urbanization had no effect on carbon signatures, which reflected corn, or the concentration of fecal antioxidants. Based on my results, chipmunks in urban areas may be consuming more protein from bird feeders or food waste, and may be experiencing metabolic consequences associated with a high protein diet compared to their natural counterparts. For example, prior research has shown that when mammals consume lots of protein resources from meat, they may be at risk for high cholesterol, which may put them at risk for cardiovascular diseases. One thing that I found surprising within my results was that carbon did not differ between habitats, and that might be because of locations. So for example, my gradient was relative to Sudbury city limits. Had I included other study sites that may be more natural, I may have seen different results. Or it's possible that chipmunks may preferentially choose food types, so they may be avoiding foods that are high in carbon for whatever reason, maybe because they have access to compost bins, which may have better resources for them relative to bird feeders or garbage that have carbon waste in them. Chipmunks did not differ in fecal antioxidants, which may be because they're consuming similar amounts of antioxidants on the gradient. A typical diet that a chipmunk would consume in their natural habitat consists of vegetation, small arthropods, and bird eggs. So it's possible that those resources are providing them enough antioxidants that we wouldn't see differences between chipmunks and urban habitats that would be consuming human food waste, fruits and vegetables out of our garbage bins and compost bins. It's also possible that the spectrums of the gradient did not differ enough to see differences in antioxidants. Had I considered other, maybe more natural study sites, we may have seen a difference. As well, many chipmunks that I caught in backyards had access to forested areas behind the property, so it's possible that chipmunks may be spending time in both more natural areas and more urban areas. To summarize the meaning of my research, chipmunks may be experiencing consequences depending on the diet that they consume, which may put them at risk of metabolic diseases such as high cholesterol and cardiovascular disease. A human diet may influence reproduction, so because we know these chipmunks are consuming more human food than their natural counterparts, they may be able to have more bouts of reproduction or perhaps more offspring. And isotopes may distinguish individuals living in urban and natural environments because we know there is differences in the signatures across individuals. My research helps us understand how other animals may be responding to growing rates of urbanization and the access to human food waste, so we can better understand how to deal with the physiological consequences that these animals may be experiencing. Thank you.